One of the major diseases of today is diabetes. And diabetes is a disease that is spreading all over the world as an epidemic. And it's also going down in age groups, which basically means that kids are very often today getting this disease.
That is why we have now this enormous increase in type 2 diabetes, is because we take very good care of our cars, but we take extremely bad care of ourselves. We just load us with whatever kind of junk we can find.
Obesity is going up. It is the lifestyle and its genetics that make this happen.
Fat is the most energy-containing substance that we have. And if you want to eat high-fat diet, then you should have a high energy expenditure. Otherwise, you shouldn't eat so much.
The disease is a disease associated with destruction or malfunction of the cell that is secreting insulin, which is a major and important hormone for regulating the sugar concentration in the blood. And if this is not working, we get too high levels of sugar, glucose in the blood, that leads to a lot of complications in important organs like cardiovascular organs, like nerves, like kidneys, like eyes.
The Rolf Luft Center at the Karolinska Institute in Stockholm is a research center for diabetes and endocrinology, focusing on diabetes and its complications, as well as tools that enable validation of novel targets and drug candidates for the disease. Head of the research group is Per-Olof Berggren. Both early and applied research is executed at the Rolf Luft Center, as well as in satellite labs abroad, in Singapore, Miami, and Korea.
My research over the years have been focused on understanding why the insulin-secreting cell is not working.
A unique way of studying insulin regulation in the living body has been developed at the Rolf Luft Center. Inside the body, in the pancreas, the factories for glucose-regulating insulin are situated. The islets of Langerhans, small bubbles containing thousands of beta cells responsible for the making of insulin. From the pancreas of an animal, a small sample with islets of Langerhans is removed and relocated to a very different place. With a thin tube, the islets are implanted onto the iris of the animal without disturbing the field of vision. With a microscope, the living and fully functioning islets are now easy for the scientists to study, located in the body's only biological window, the eye. The cells are taken care of by the body, with blood vessels and nerves growing into the islets. The cells continue their normal task to produce and distribute insulin.
The transplantation into eye has been known for quite some time, that it's possible. But this is the first time when it's applied in the way that we can use it for imaging.
The tremendous advantage with the InSight model is that we now, for the first time, have a possibility to study the insulin-secreting cells in the living organism and not in isolation, as we have done before.
This is a very robust system, and that's the beauty of the model. So we keep 90% of the islets in the eye, and then I wait for 3 to 4 weeks until the islets are getting vascularized and are stable and grafted onto the iris, and then we would image them.
You don't need to penetrate some tissue. You can come back the same day, you can come back the next day, the next week, the next month, and study exactly the same tissue. You can see what happens in the living organ.
We are looking at a number of signaling pathways that are then also developed into different projects. And for us, the most important thing is to understand the whole picture of how the pancreatic islet cells are functioning. We have identified 3 basic novel druggable targets that we think are important for the development of new drugs in the treatment of diabetes.
One of the projects identified as a priority investigates the death of pancreatic insulin-producing cells, why the cells die, and thereby give rise to diabetes. Evidence showed that something in the blood serum generated a lethal activity in the cell's so-called calcium channels, causing the death of the cell.
So the cells died. But if we blocked these calcium channels, the cells survived. And then, of course, the question arises, what is it in the serum that gives this effect?
When I got the material, I had to find a way to find out the factor in the serum that was doing that to the beta cells. And I realized from the start, I had enough material to know the mass of it, the weight of it, and activity.
We fractionated the sera and purified and found that the substance or the protein in this sera that gave this effect was apolipoprotein C3, which is well known about people working with cardiovascular disorders.
There are populations around the world where apolipoprotein C3 is down by 50%, and these populations are living forever. They don't get old, they don't get cardiovascular problems, and they don't get diabetes. So this is a very important target.
If you have type 2 diabetes and insulin resistance, then you also have higher levels of ApoC3. So we know it's interesting from both aspects, both type 1 and type 2. So really what we are aiming to is to get the permanent lowering of ApoC3.
If we are able to suppress ApoC3, we are not only making a big thing for diabetes, but we are also making the overall population more healthy.
In every one of us, our organs demand a constant flow of fuel in the form of sugar, glucose. To regulate the sugar level, the organs have receptors for insulin, signaling to the insulin-producing cells to increase or decrease the delivery.
Insulin in a cell is almost involved in all aspects of cellular metabolism, cell protection, the life of a cell.
At Rolf Luft Center, the research group has made an important discovery, that insulin receptors also appear on the surface of the insulin-producing cells, completely changing the understanding of insulin regulation in the body.
The findings that insulin-producing cells can sense insulin was unexpected, and still there are people out there not really believing, because it is breaking with a biochemical dogma.
This is now a second important project aimed at delivering a new treatment alternative.
The fact that these insulin receptors are also present on the surface of the pancreatic beta cell means that they are important for normal beta cell function and survival. And if we have blindness to insulin in the beta cell, we get deleterious function of the beta cell.
And what is even more remarkable is that the insulin-producing beta cell has two different kinds of insulin receptors with a tiny but very important genetic difference.
We have learned over the last 5 years that people that have to treat themselves with insulin have, unfortunately, a higher risk of developing certain type of cancers. If insulin is secreted, it binds to the A-type receptor and it binds also to the B-type receptor. That is the one who is doing the good job reducing your blood sugar levels in muscle, liver, and fat, while the A-type receptor would be the one that is promoting your cancer progression. It is time for going for a new type of strategy for treatment. And what we had in mind is to look for smarter insulins that goes for B-type receptors and not for A-type receptors.
And this is also one question we are actually trying to focus in Biokine on. Trying to find some partners that can, with us together, develop this compound. But it could be also something different, where you just block insulin receptor A and then allow only the B signaling, giving the metabolic actions.
A third novel druggable target is the study of a calcium channel subunit inside the insulin-producing cell.
The voltage-gated calcium channel is a miniature calcium conducting tunnel with an electrical gate. Such a mini tunnel is made up of 4 or 5 different protein building blocks. One of them is called beta-3 subunit.
We are very interested in a protein that is transporting calcium into the cell and of components of that channel, because we have found that if we are manipulating components of that channel, we can also enhance glucose-induced insulin release. We may find a small molecule, a small compound that is blocking this subunit, and by blocking this subunit, we will get a beta cell that is now much more willing to release insulin at a high glucose concentration. So that will be exactly a compound, a medicine we are looking for. In addition, blocking this subunit will lead to better survival of the insulin-secreting cell and also more production of insulin.
The scientists working at the Rolf Luft Research Center and at the satellites abroad represent many countries around the globe and are among the most experienced in the field.
We have several subgroups in the group of pyroberberine, so it is possible to have an intergroup communication and contribution from different people to different projects, so this makes it a very fruitful atmosphere.
Weekly video conferences with team members in Sweden and the satellite labs abroad coordinates and updates the efforts within the different fields of research.
Within the Berggreen lab here, we have people coming from Russia, from Germany, obviously. We have people coming from Great Britain. We have
Italy,
people coming from Italy, from China.
Yeah.
So, and we have even some Swedes here.
Since the major treatment of diabetes is physical activity, it's very important to keep fit as much as you can, and therefore physical training is something that everyone should actually be involved in. It's fun. We have done that for a long time, and then you feel better, and hopefully you live longer, and you live a healthier life.
It's always challenging to work in different disciplines. And PO has managed to gather a group of highly qualified different disciplines in the lab, which, when they integrate with one another, they produce great science.
So, among the different leads towards new possible treatments for diabetes developed at the Rolf Luft Research Center, the most promising are lowering blood levels of ApoC3, modifying activities of insulin receptors A and B, and blocking a voltage-gated calcium channel subunit beta-3.
And recently, our major achievements have been that we can study the cell in the living organism. We have also shown new treatment strategies for this devastating disease. And this is extremely important, as I said, because it's not only that this is very complicated and painful for the individual who gets the disease, but it is also associated with enormous costs for society. So if you put these two things together, we need to do something about diabetes at the end of the day.